I want to start with this very basic idea that actually goes back to the middle of the last century, that actually has strong roots in Israel, which is this notion that a very familiar enzyme, acetylcholinesterase, actually can have a second function that has nothing to do with the hydrolysis of acetylcholine. Going back into ancient history, it was first mooted in 1969. It was the subject of a chapter of a book in 1974. I came on the scene exploring the concept in the 80s. And then I'd like to pay tribute to Mona Sarek, who I'm sure many of you know at the Hebrew University, who's been a pioneer and is a very dear and close friend who's worked on this for a long time. So this idea that cholinesterase can have novel functions is rooted in various findings over the years. First, it's present in areas where there's no cholinesteryltransferase or acetylcholine, and indeed in non-neural cells, so it doesn't have to function as an enzyme. We've shown that it's released as a signaling molecule in a calcium-dependent potassium-overt way. We've blocked the site irreversibly with SOMAN or sarin and shown that it can still have a range of electrophysiological, biochemical, pharmacological, and behavioral actions. And that action is to promote cell growth via, perhaps very unoriginal, the entry of calcium. So this might be a very important but as yet overlooked process in cell growth and renewal, and therefore have implications not only for pathology but for aging generally. And it really raises the question, what's the salient part of the molecule? Because of the shortness of time, I'd like to summarize about 20 years of research by giving you the answer and introducing you to a 14-mer peptide called T14, which originates from the C-terminus of acetylcholinesterase, has a partial homology, as it happens, with amyloid. And as you can see here, the 14-mer is the active sequence. We also sometimes use a 30-mer, which encompasses the 14-mer, allows us also to have the 15-mer at the C-terminus, which we use as a control. We've isolated and identified this as a salient sequence that underlies non-cholinergic functions. And what we've shown is it promotes cell growth. So here you can see in the developing rat brain, in the embryo, it's the highest, declining through into maturity. That is mirrored, that qualitative measurement is measured quantitatively in the developing rat brain. You can see here P7 being the highest. That in turn correlates with optical imaging of large groups of brain cell activity that we call assemblies, which reflects the depolarization of cells with fluorescent optical imaging. And then in collaboration with George Mason University, you can see here in shy cells that the peptide has enhanced the surface area of the cell and indeed shown that this is probably due to the activation of intracellular mTOR because it's sensitive to rapamycin. We picked that idea up and showed similarly that in the human brain that mTOR correlates closely with T14. And then if you look in the brain, you can see with normal aging, this is post-mortem human tissue, there is decline. What's very interesting is the provenance of brain cells is the same as for skin cells, and we've just been hearing about skin. We were therefore interested in looking at the role of T14 in keratinocytes in the epidermis, and we did this in collaboration with Unilever. And you can see here the 23-year-old, the T14 positive keratinocytes in the epidermis showing very strongly and declining rather depressingly into the 50th year. We've also done pilots on sebum and saliva and shown a similar pattern of an age-dependent decline. So we thought this would be of interest to this meeting because clearly there might be something here to do with development and aging encompassing this previously little-known 14-mer peptide. But the problem is an agent that drives cell growth can also turn toxic. This will depend on amount, as shown here, in this case with N-methyl diaspartate a long time ago, looking at neurite outgrowth, where you can see with increase in concentration, one goes from trophic to toxic. That is also the case when T14 is applied in a rather Jekyll and Hyde type way, where you can see the enhanced neurite outgrowth with the low levels of T14, whereas as you increase the levels, then the cells are severely compromised. Or you can have duration of application where for a small amount, a short period of time, there's an enhancement of neurite outgrowth, whereas if you keep the same amount on for longer, then it actually turns toxic. But the third and most interesting way in which you can have trophic turn to toxic is actually from Emel and Schramm, a very good paper showing that with age, the same amount of calcium will be toxic rather than be promoting cell growth. So this struck us as an interesting idea that perhaps this is what underlies neurodegeneration, and perhaps one could think of Alzheimer's disease and related conditions as an inappropriate form of development. Again, time restrains being able to give you all the evidence, but just some of it is, if you look in Alzheimer model mice, you can see in the transgene negative adult, there is very little signal shown here in A, whereas in B, C, and D, in the adult, you can see T14 immunoreactive plaques showing that it is a hallmark of pathology. If you then look in the rat brain, as we've seen, it declines with age, and this is recapitulated in the human midbrain, which is an area containing the primarily vulnerable cells in Alzheimer's, which is not the hippocampus or the cortex, but actually subcortical structures encompassing the midbrain and brainstem. And you can see here it's almost doubled in the postmortem tissue. To cut a long story short, again, because of time, we've then identified the receptor and that the T14 works on, and shown that it works exclusively on the alpha-7 nicotinic receptor at an allosteric site. First, because in oocytes, only those that are expressing transfected with alpha-7 is there a response to the peptide. Both are highly co-expressed and correlating with human hippocampal tissue in patients with Alzheimer's. Alpha-bungotoxin will have selective block. We can see increases when we either mutate or overexpress the alpha-7 receptor. And we've shown that it only affects responses in areas that are rich in alpha-7, and that excludes the stratum, which, although it contains cholinergic receptors, doesn't actually have the alpha-7. And moreover, we've shown that the peptide can enhance, it can actually enhance its own target, as shown here in Schei cells, and indeed in GH4 cells in our own hands. So quite unusually, this would be a rather tempting target for Alzheimer's disease, given that it seems to be exclusively at this allosteric site, as shown here. So what we've done is actually fight like with like, and unlike a lot of attempts at blocking this receptor that have been tried in Alzheimer's disease, what we did was to fight like with like by cyclating the peptide, as shown here, and shown that it actually does bind to the site of its linear counterpart, displacing the toxic endogenous agent, as shown here in ex vivo brain slices, where we have severed the midline and incubated one side with the drug and one side without. We call the drug NBP14, neurobiopeptide 14. Here you can see it given a single dose in vivo into the living animal, where it's maximally displaced at 4 hours. And then in the human brain, you can see, if you look at the binding of alpha-7 and the T14, that this can be intercepted in a dose-dependent way by NBP14. So it does seem that that is the way it's working. The question is, does it have any effects? And in the 5-fat animal mouse model, and I know there's constraints on the animal model because there's no authentic animal model as yet. Nonetheless, in such model mice, given intranasal NBP14 twice a week for 14 weeks, we can see a healthier weight gain compared to the controls that becomes more emphatic over time. And if one looks at novel object recognition, where a rodent will explore an object for a longer period of time if it's novel, you can therefore use exploration time as a way of finding out whether the animals remembered or recognized an object or not. You can see here in wild type animals, the red being, in this case, the novel object, much more exploration time has occurred for that. In the saline mouse model, the vehicle, the mice can't distinguish novel from familiar, whereas in the model, given our drug, you can see their performance is comparable to the wild type and that they can distinguish very well familiar from novel. This is emphasized by also looking at amyloid, which we see as a downstream agent, not the primary target of Alzheimer's, but a downstream agent that's indeed triggered by our peptide. And you can see here the gold blobs being the amyloid, that indeed in the NBP14 treated animal, there is far less amyloid. It's blocked the production of it. So given that, it seems that T14 is indeed an agent driving cell growth and renewal with applications to Alzheimer's. It continues in all age in the skin, but as well as if it's aberrantly activated leading to neurodegeneration, it can also, we would argue, lead to metastases. A definition of cancer is, after all, an inappropriate activation of developmental mechanism, or indeed to skin disorders such as eczema and psoriasis, which are hyperproliferative keratinocyte disorders. So in one slide, just to summarize where we are with that, this we published back in 2017 with myeloma, leukemia, and breast cancer cell lines, where we compared metastatic cells with non-metastatic cell lines. And you can see in this particular case, the red is the MBP-treated condition, and that has reduced the cell migration in the strongly metastatic breast cancer cell line to that of the non-metastatic cell line. And we are now seeing encouraging results in xenograft animals for both breast cancer and colorectal cancer. The skin we saw just recently, was age dependent, and you can see here that not only is it age dependent, but it's accelerated with photoaging, showing that it's not just related to age, but more to exposure of the skin to the sun. So it does seem to be a very sensitive index of that. We mentioned previously that the mTOR was activated by the peptide, and that's blocked by rapamycin. Rapamycin, which is a drug now of increasing interest to people for aging, can compromise cell proliferation, whereas our MBP14 does not compromise cell proliferation at all, and yet it has a beneficial effect. As you can see, the peptide enhances mTOR in an MBP14 sensitive manner. So therefore, we are suggesting that perhaps our drug could eventually be preferable to rapamycin, because rapamycin presents with these side effects, anemia, low platelets, diabetes, due to the compromised cell viability, whereas our drug would apparently not do that. So to summarize, we're suggesting that with normal aging, T14 is not just in the brain, but throughout various parts of the body, declining with normal aging. If it's inappropriately activated, then it can lead to a variety of problems, from skin disorders to cancer to Alzheimer's and related neurodegenerative diseases. So in the ideal world, in the long run, the dream would be to get the correct balance of T14 in these tissues. Toda rabba.
Toda rabba. Any questions?
With the microphone, please.
Anyone? Wonderful talk. Thank you so much.
Okay, thank you.